Not seen anything. José tá sem som.
So it took some time, huh? I don't see you, but it doesn't matter.
Estão me ouvindo? Olá, estão me ouvindo? Ah, estão ouvindo? Vamos começar, então. Bom, pessoal, boa tarde. Estamos dando continuidade à nossa excelente semana de pós-graduação em Química da UFSC, em sua 10ª edição, em comemoração aos 50 anos de nosso programa de pós-graduação em Química da UFSC. Na tarde de hoje, teremos o prazer de ter conosco o professor JEAN MARIE LEHN, da Université de Strasbourg, e do ISIS, Institut des Sciences et de l'Ingénierie Supramoléculaire. Bonjour, Jean-Marie. Est-ce que vous allez bien? Bienvenue.
Oui, bienvenue.
Oui, tudo bom.
Nice to be in touch.
Tout d'abord, nous tenons à vous remercier d'avoir accepté notre invitation à prendre la parole lors de notre événement. Vous avez toujours été un grand ami de la communauté scientifique brésilienne et nous sommes très honorés de pouvoir vous accueillir parmi nous. Je vais maintenant faire une très brève présentation de vous en portugais à l'auditoire qui nous suit et je vais vous donner la parole juste après pour que nous puissions entendre votre conférence.
Très bien.
Eu fiz um agradecimento ao Jean Marie e comentei que ele sempre foi um grande amigo da comunidade científica brasileira e que nós sentimos muito honrados de tê-lo aqui conosco presente para fazer uma apresentação em nosso evento. Sobre o nosso convidado, Jean Marie Lehn é um cidadão francês que tem muito orgulho de ser da Alsácia, a região em que nasceu, no nordeste da França, mais precisamente em Holstein. Filho de Pierre e Marie Lehn, seu pai, padeiro e também organista, transmitiu a Jean-Marie gosto pela música, que vem a ser seu maior interesse depois da ciência. No campo da química, fez o seu doutorado com o professor Guy Risson, em Estrasburgo, sobre a conformação e fisicoquímica de terpenos, usando pioneiramente o primeiro espectrômetro de ressonância magnética nuclear do seu laboratório. A seguir, estudou com Robert Woodward, Prêmio Nobel de Química em 1965, em Harvard, participando do grande empreendimento que foi a síntese da vitamina B12. Retornando a Estrasburgo, em 1965, montou seu laboratório, onde, com seus estudantes, tem, há 56 anos, desenvolvido pesquisas na vanguarda do conhecimento. Paralelamente ao seu trabalho em Estrasburgo, em 1980 foi eleito professor no prestigiado Collège de France. Jean-Marie cunhou o termo química supramolecular, estabeleceu as bases para a área e, ao longo do período, fez nascer e viu florescer dezenas de linhas de pesquisa que despertaram interesse na química supramolecular em todo o mundo. As centenas de estudantes formados em seu laboratório espalharam-se por todo o mundo e trabalham como professores e pesquisadores na indústria em diversas universidades. Sua prolífica produção científica, na forma de livros, capítulos de livros e artigos científicos, mais de 1000, confere ao seu trabalho índice H maior que 150, segundo o Google acadêmico, e de acordo com o Web of Science, índice H139, com mais de 87.000 citações. A sua carreira científica tem sido reconhecida e premiada com centenas de distinções, dentre elas o Prêmio Nobel de Química de 1987, dividido com Charles Pedersen e Donald Cram, e na forma de doutorados honorários concedidos por mais de 60 universidades de todo o mundo. No entanto, o que eu gostaria de reforçar aqui são algumas particularidades que marcam a personalidade de Jean-Marie, que pude verificar no período em que tive a felicidade de trabalhar em seu laboratório. Ele tem um conhecimento enciclopédico de química, uma memória prodigiosa e uma vasta cultura. Tem uma enorme capacidade para conseguir amarrar com facilidade diferentes áreas do conhecimento. É capaz de fazer exposições muito didáticas de assuntos muito complexos. É grande entusiasta da multiculturalidade, o que é comprovado pelo fato de que estudantes de todos os continentes sempre trabalharam em seu laboratório. Eu me lembro de uma festa de fim de ano do laboratório em que ele se orgulhou de ter ficado muito feliz ao ver que estavam presentes estudantes de mais de 18 países. Considerando-se este aspecto, ele sempre deixou as portas abertas para nós do Brasil. Daqui da UFSC, que eu fiz pós-doutorado em seu grupo, e atualmente Paulo Costa, que fez doutorado em nosso programa, está em seu grupo também fazendo estágio de pós-doutorado. É físico-químico orgânico de formação, que imprimiu desde o início ao seu grupo a ideia de buscar sempre estar à frente das fronteiras do conhecimento. Além disso, é um entusiasta pelo trabalho árduo, disciplinado e organizado, e pela generosidade em ciência. JEAN MARIE trabalha todos os dias, incansavelmente, de segunda a domingo, sempre se entregando apaixonadamente na tarefa de ensinar e fazer ciência. No período em que trabalhei com ele, todo mundo dizia que ele trabalhava o tempo todo, mais que todo mundo, e também no Natal e no Ano Novo. Diziam também que se alguém estivesse trabalhando no Ano Novo, ganharia dele uma garrafa de champanhe. Eu não acreditei de início, mas fui ver se era verdade. No Ano Novo estávamos apenas eu e ele trabalhando no laboratório. Ele veio me cumprimentar. Só ficou devendo champanhe. So, JEAN MARIE, thank you again so much for being here with us. Please feel free to make your presentation.
Okay. So, for me, it's also a pleasure to be in touch with Brazil, with this university, and of course, to see again my former collaborator, Vanderlei. Long time we haven't seen each other. So it's a pleasure to participate in this anniversary celebrations, 50th anniversary of your program in chemistry at the postgraduate level. I have chosen a general presentation, but I have added some more chemical parts, also sections, so that there is some more chemistry in it. The title of my presentation is Steps Towards Life. This does not seem to be chemistry, but I would like to show you that life has a lot, if not everything, to do with chemistry. For doing that, we have to start very early on, very, very early, for 13.7 billion years ago, with the Big Bang. Then our universe was born. But the universe was very, very hot. No chemistry. It was very hot, it cooled down, particles formed, and the particles then got together to make atoms. That is the age of physics. Then the atoms connected to make molecules. And molecules evolved also, became more and more complex, and became different, very different combinations of atoms. That's the age of chemistry, which starts around here. But these molecules formed in our universe then continued to become more complex. They began to associate together to make assemblies of molecules, compartments, and a new property appeared, which was life on our planet around that place. This is the age of biology. But on our planet, life continued to evolve and generated another property, a very important one, thinking, thought. And this is steps towards life and thinking, of course. Let me just point out that here you see the end of my screen, but that is not the end of the evolution of the universe. The universe will continue to evolve. It will generate other entities. We are now part of it, and we can try to understand what is going on. So we have to take into account something. This is a real slide, because I want to show that in our universe, according to cosmologists, there is right now 68% dark energy, 27% of dark matter, which means 95% of darkness. However, the 5% left are 5% of visible matter, and that is the matter that matters. That is our matter. We as human beings and all living organisms on other planets, and there are certainly other planets with organisms with very probably life, they are part of this 5% of visible matter. So in the course of time, matter started by being divided, then condensed, then organized, living, thinking, and maybe something else beyond that, something even more complicated than our thinking. So matter became more and more complex. It contains more and more information as time goes. We then have to ask, a big question. I consider it as probably the biggest question in science. How does matter become complex? How can one go from elementary particle to a thinking organism, and maybe even to higher forms of complex matter? To give an answer to this big question, mankind has created science. And science has several fields, pillars. Physics deals with the general laws of the universe on which everything depends. Biology deals with the rules of life. And chemistry has the mission to try to build the bridge between the general laws of the universe and complex matter as it is expressed in biology, in life, and in thinking. This is, of course, an enormous project, an enormous duty to do that, and the mission. And the answer to the question we may consider is self-organization. What I mean by that I personally think, and I'm not alone, that our universe has a structure such that self-organization will occur and will lead to complex matter, including life and thinking. One may even say that it is a cosmic imperative. The structure of our universe is that it will generate living and thinking organisms, and it is probably been happening in other regions of the universe. So self-organization, we have to look at it in two ways still, because there's no unified theory yet. Cosmic self-organization deals with the cosmic structure of the universe, and this is organized through gravitational forces which act on the inhomogeneities in density and in rate of expansion right after the Big Bang. But this is not what we are interested in. It is, of course, very interesting, but it doesn't concern us so much. Complex matter is what concerns us. Molecular matter is formed through electromagnetic forces acting on the bricks, on the pieces, on the elements which make up our universe, leading to organized, living, and thinking matter. So the chemical evolution of matter started on our planet much before life was existing. So prebiotic chemical evolution deals with the self-organization of non-living matter. And later on, the electromagnetic forces in our universe operate selections on structural diversity with formation of more and more complex entities made from the bricks which form visible matter in our universe. Once life has appeared, Darwinian evolution is starting, the evolution of living organisms, but even that is also chemically based because, as you know, Darwinian evolution rests on the transmission of information through generations, and this depends on DNA, and we will come back to that, which is a chemical entity, of course. So usually one describes chemistry as the science of the structure and transformation of non-living and living matter. This is correct. But we will see that there is another feature of chemistry which is very, very important. We see in a moment. Let's go back 2,500 years to have an early view of the structure of matter as seen by Greek philosophers. Empedocles, about 2,500 years ago, considered that the universe, that matter was made of 4 elements: fire, air, water, earth, which led to properties: dry, hot, humid, and cold. It's a start, but it is really rather simple. However, at the same time, more or less, another Greek philosopher, Democritus, said that matter is particulate. It is made of pieces which cannot be made smaller, atomos in Greek. And that was the start of the name of atoms. So Democritus had already the sense that matter was made of particles, and these particles were the elements which build up visible matter. Now, over the years, scientists, that means people who were philosophers and analytical people, or all kinds of wise persons who wanted to learn about structure of a universe, ended up by finding that the universe contained things which one could isolate, which were called elements. But these elements were not well organized, and there was no general rule for that until the middle of the 19th century, when several proposals were made for the structure of these elements and how they could be organized. The most important one is Dmitri Mendeleev, who in 1869 published a very famous and extremely important paper, which you see here. This is the original paper of Dmitri Mendeleev, published in 1869 in the German journal Zeitschrift für Chemie. It's about 150 years now since then. And here you see that the title in German is in English, On the Relationships Between Properties and Atomic Weight of the Elements. Using these two parameters, he could organize the elements in a table which you see here. That's the original table of Dmitri Mendeleev. And you see that there are columns, there are lines. Of course, right now it's a bit differently organized, but the table is correct. The way it is organized is correct, although the columns have now become lines and the lines have become columns, but it doesn't matter. Furthermore, Mendeleev was convinced that when some elements were not present yet, as you see here, there are question marks in the table, he said that these elements will be discovered, and of course they were discovered. So I consider this as one of the most important publications in science. This is the publication which organizes the elements in our universe. And nowadays, this periodic table of the elements is now this one, as you see it here. All of you know that, I suppose. All the chemists must know this table. But nevertheless, I would like to insist on it, because looking at this table, you see the elements which make up visible matter. This table is complete. There are no others. Between carbon and nitrogen, there is no other element. Between nitrogen and oxygen, there is no other element. So the table is complete. I find that an absolutely incredible realization achievement, that mankind on our piece of dust, the planet Earth, has been able to determine what visible matter is made of. And I insist, there are no other elements. If there are living organisms on a very far planet, they will be made of the same elements, maybe somewhat different than ours, but it will be the same elements. Hydrogen is the same on our planet at the end of the universe. Carbon is the same on our planet at the end of the universe. So this always, I'm amazed, it always, in admiration and amazement that we can have before our eyes the table which represents the bricks, all the bricks of our universe, of the visible matter in our universe. And this is the playground of chemistry. Chemists play with these elements, like with bricks when you build a house. They play with these elements to construct chemical objects. Of course, there are chemical objects which already exist, but you can, at will, recombine these, following, of course, the laws of physics, which allow some things to happen and not others. Now, this is the structure of matter. The transformation of matter was first described in general terms by Lavoisier in France, when he wrote, "Nothing is lost rien ne se perd, nothing is created, rien ne se crée, tout se transforme, everything is transformed, meaning that when you have a set of elements making up some part of matter, then you can reconstruct it, you can recombine it, transform it, but it will not disappear, it will not be lost. Now, this chemistry, therefore, has a composition of matter, it has a transformation of matter, but is that all? People noted then in the bricks of visible matter, there were connections between the elements. But so that it's a sort of a graph, but it's not just a graph. So matter is not composed of entities which are just graphical. They have an architecture. And in the 19th century again, a little bit after the middle of 19th century, it was proposed independently by two people, two chemists, Van 't Hoff and Nobel, that molecules, chemical entities had an architecture, and that is the start of stereochemistry, chemistry in space. So from the atom to the molecule, you know, I could spend much more time, of course, on the history of chemistry. That is sure, but this would take a week or a month. But let's now, just with these few examples I have given, from the atom to the molecule, molecular chemistry started by combination of atoms to make molecules, mastering the organization of molecular matter. Now, if we want to look at some milestones, we can consider as milestones in molecular chemistry, first of all, the fabrication in the laboratory of urea by Friedrich Wöhler in Germany. Wöhler produced urea from an entity, ammonium cyanate, in the laboratory. Ammonium cyanate is not contained in living organisms. Urea is, of course, contained in urine. So, by doing so, Wöhler showed that between the molecules of non-living matter and the molecules of living matter, there was no difference in principle. That also destroyed the idea that to make anything contained in a living organism, you need this magic force called the vital force. Now let's go further. 150 years later, the following molecule, vitamin B12, was made in the laboratory by the common efforts of two groups, one directed by Robert Burns-Woodward at Harvard, the other one by Albert Eschenmoser at the ETH in Zurich, in Switzerland. They were, of course, helped by many collaborators. It was a collaborative effort with many people, many researchers, usually mostly postdocs, but not only postdocs, graduate students, PhD students, postdocs. And together, they succeeded in 1972 to make this very complex molecule, vitamin B12, from its elements. This was a great achievement, and it was sort of considered as the pinnacle of molecular chemistry, showing the power of molecular chemistry. And of course, it continued in the years after the 1970s, and many new reactions were invented, new products were made, new industrial processes were developed, and materials and all that, drugs. So molecular chemistry continued very actively to develop, and it will continue for many years. But then we can ask a question, is there another part of chemistry we should be looking at? I can illustrate that by showing you here a cancer cell in blue and two killer cells in purple. The killer cells have a mission. They must find out whether the other cell is a cancer cell or not. That means a cell which is transformed or not, and they have to destroy it. But they should not make a mistake. Killer cells must destroy the bad cells, but not the good ones, the healthy ones. Similarly, here you see the white blood cell with blue dots, which is the HIV virus. When the HIV virus gets in contact with a white blood cell, it can infect it. What tells the virus that it has reached its goal and can now infect? So this means that between these bodies here, the killer cells and the cancer cell, the virus and the white blood cells, something happens which tells each partner what the other one is. Killer cells can know that the other one is a cancer cell thanks to interaction between molecules present in their membranes. So that means that beyond molecular chemistry, there must be a chemistry which deals with assemblies of molecules, a chemistry which lies beyond the molecule and which I have termed supramolecular chemistry, and which deals then with the interaction of molecules with one another, recognition between molecules, like the killer cells must recognize cancer cells, reactivity of molecules, and transport of one molecule through a barrier by another one. Molecular recognition is, of course, the basis. Molecular recognition is what happens when killer cells recognize a cancer cell. And this is by contact between the molecules. Now, we can then try to see chemistry as in 1828, molecular chemistry based on single molecules and the structure of molecules, making by synthesis receptor molecules. And in 1978, I proposed the name of supramolecular chemistry for this part where the molecules are not anymore considered as isolated, but they are together in a population, in a mixture, in assemblies of molecules. And this then leads to three possible features: recognition between molecules, transformation of molecules, reactivity, and translocation transport, leading to functional supramolecular system. And these features were all studied very extensively by many, many laboratories around the world. So my initial motivations for this field were the following. I was interested in neurochemistry. Now you can say, what has neurochemistry to do with this? It has to do the following. The propagation of the nerve influx is a rather simple, at least the propagation of nerve influx rests on a rather simple events in biological systems. It is relying on an action potential, which is caused by exchange of sodium and potassium ions across the nerve membrane, and which then propagates. And this carries the nerve signal along the axon of the nerve. So here was something which chemists could study in the 60s, because sodium and potassium, of course, are very simple chemical entities, and maybe one can then try to understand how one can selectively bind and transport sodium and potassium ions through the membrane of the axons. There must be molecules in this membrane which are able to make the difference between a sodium ion and a potassium ion. Here are the series in the periodic table of the elements, the column of lithium, sodium, potassium, rubidium, cesium, the 5 alkali metal ions. I don't talk about francium, which is a radioactive one. So in this series, the 2, sodium and potassium, are biologically very important, and they are a series of spheres carrying one positive charge. So how can the selective binding and transport of spherical ions be generated? Is there spherical molecular recognition, and can one understand it? we made at that time in towards the end of the 1960s, we thought that one way of which may lead to the recognition is to synthesize molecules which contain a cavity which is compatible with the size of a given ion. So one can make what we call cryptons because they have a cavity inside the molecule, and one can synthesize those in the laboratory, and by chemical synthesis adjust the size of the cavity to the size of what one wants to put into the cavity. So from here to the middle and to the right, the cavity is compatible with lithium and sodium and potassium, and this indeed functions, and these cryptons lead to selective formation of the corresponding cryptates. Now, this type of molecular recognition, in fact, looks like the fitting together of two chemical entities, the receptor unit, which has a cavity, and the objects which will be entering into that cavity. So one can then look at how to represent in very simple terms molecular recognition. Molecular recognition requires an interaction for binding. You need to bind objects together. If they don't bind, they ignore each other. But that's not enough. You know very well that if you want to recognize somebody in the street, you need to have information. So it requires also information for performing selective binding, not just binding, but selective binding, selecting. And this resides, in simple terms, in a double complementarity of geometry and interactions. And it was already proposed many years ago, although at that time it was very mechanical. It was not like nowadays, more interactional and physical. And this was proposed by Emil Fischer in 1894, when he said, and for an enzyme to act on its substrate, they have to fit together like Schloss und Schlüssel, like a lock and a key. This is a very simple idea, but it is a very strong image, which fits more or less well the reality. Molecules have to fit together, and that is molecular recognition. Now, the most important process of molecular recognition is the one which is, of course, the molecular program, first of all, for storing information in living organisms. Living organisms are defined by their genome, which is a chemical object based on a long string of rather simple units, phosphates and sugar here, ribose or deoxyribose. And on this long string are fixed letters for different chemical groups, simple chemical groups, adenine, guanine, thymine, and cytosine. And then two such strands can wrap around each other, and they form the very famous double helix of DNA. Now, these four letters in a given sequence define the genome of living organisms on our planet, and it's the recombination of these letters which makes a difference between an organism like a tomato or an elephant, a plant and an elephant. Now, this is the storage of the information which determines the living organism, the entity which will be formed. How does one recognize it? First of all, since I have talked about the steps towards life, let's just look at one of these letters, adenine. Adenine, you see here, is one of the four letters, and it is just a polymer, a pentamer. 5 HCN molecules make adenine. As you can see, HCN here, HCN a little bit rearranged, but 5 HCN give a molecule of adenine. It's a very simple process, and of course, one can easily imagine that in prebiotic conditions, HCN exists in the universe, then HN molecules could get together and progressively build up one of the letters of our genome. Adding some water gives another letter, guanine, and making some other transformations gives the last ones, uracil and cytosine. So now, how do we read this information? Of course, storing the information is one thing. But in addition to storing, one has to be able to process the information. And this is a very simple process also. It is a pairing up of two letters. Adenine pairs with thymine or uracil by 2 points of interaction, as represented by these 3 dots here. And guanine can pair with cytosine by 3 points of interaction. So in fact, it's a binary system with 2, 3, 2, 3 types of interactions. It is now remarkable and in fact sort of astonishing, but understandable, that the genome of all living organisms is just made of 4 letters in a very specific sequence, and this genome is processed by a binary pairing, 2 points and 3 points of interaction. Why is it so simple? It's difficult to say, but one can make the assumption that probably it is so simple because it's good enough to have 4 letters and 2 pairing ways to store the information and then to process it. Probably in the course of evolution, maybe other letters have been tried, but it became probably too complicated with too many errors, so that evolution started and stayed with what is working. Now, chemistry is therefore also an information science. Molecules store information, and it is between molecules that the processing of information happens. DNA, the genome, is the molecular storage. The way these interactions between the nucleobases occurs is the processing of information. So chemistry is not only the science of the structure and transformation of matter, is also a structure of information in matter, of informed matter. And that's a very important property, which in present ages, where we have more and more devices handling information, one should not forget that a living organism is a much more complicated system, and in its functioning, in its storage of information, in its functioning, than the most expensive computer. Now, molecular recognition then, in the form of locks and keys, like Emil Fischer proposed it, is shown here. We have on the left-hand side 3 keys, in the middle a lock, and of course the red key is the one which fits. So one can say that trying to understand molecular recognition is to make locks for keys or keys for locks. And this has been done in very many laboratories which have studied molecular recognition over time. What are the possible applications in life sciences of that? Because obviously, molecular recognition is an extremely important process in life. So what applications can one gain from acquiring knowledge by basic research on this problem and then applying it? First of all, obviously, drug discovery depends on molecular recognition. One can say that a synthetic molecule is a key for a biological lock. The key has to fit the lock, and then it can do something to that lock, to that biological target, usually a protein, and then this can lead to activation or maybe repression. Among the objects we have made over time, I will only give you some of our examples of applications, but there are many, many others. So here is the development of a medical diagnostics apparatus, an immunoanalysis system, where we made a cryptate of a molecule which is able to absorb UV light, which will pick up a europium ion, and when UV light is transferred, UV energy is transferred to the europium ion, it will emit a red luminescence. And this is used as a label for medical diagnostics. A transport process is gene transfer. Gene transfer is a process by which a piece of DNA, a gene, is introduced into a cell by a synthetic vector or also by viruses, and then goes to the nucleus, generates RNA, and finally the product, protein. This is a very important process, very important for gene therapy to correct for genes which are non-functional in an organism, in a human being, for instance, and also for biotechnology to modify organisms. Here, I would like to insist on the fact that genetically modified organisms obtained by gene transfer are very important. GMOs, the process of making biotechnologically genetically modified organism, is very important for mankind. We have to develop it. We have to use it. I am convinced we will use it. There are people afraid of it. That is too bad for them. But I think we have to use genetically modified approaches for creating, for instance, plants which resist high temperatures. Then another interesting application was the generation of supramolecular materials, especially supramolecular polymers. And let me just give you an example of what has been done in this area. It's an area by itself, but let me just show you. In 1990, we published that paper which defined supramolecular polymers and introduced the concept of supramolecular polymers. Now, can one make biomaterials out of supramolecular materials? Indeed, one can. And in 2013, the first worldwide application of biocompatible polymers was done. This is for cardiovascular implants in children. A small company, Xeltis, developed supramolecular polymers, biocompatible ones, for the surgical treatment of children having a very severe cardiac malformation and needing cardiac reconstruction. This was done in October 2033, and here is the first child which was implanted, Dominica, at that time 4 years old. In 2013, now she's 11, between 11 and 12, I suppose, now, yeah. And this is Professor Leo Bokiria, the surgeon who, at the Bakulev Center for Cardiovascular Surgery in Moscow, implanted Dominica. This was for us a fantastic satisfaction, of course, to see that compounds, a concept which we had proposed, compounds which had been made, but of course they had been developed by Xeltis, by the company, in other means, and for instance also thanks to the work done on supramolecular polymers by Bert Meyer, professor in the Technical University of Eindhoven in the Netherlands. A young girl was repaired, so to say, had her heart reconstructed thanks to these polymers. Many children now have been treated over the years, and now the same company has also made heart valves which have been implanted in children in Budapest, in Krakow, and in Kuala Lumpur. So this was an extreme satisfaction for us to see that basic research had led to a device which was now used in human bodies in children. It took 23 years to do so, from 1990 to 2013, but it happened. And of course, as you know, introducing something in a human body, it takes a lot of, one has to be very careful, a lot of care, but it happened, and it just takes time. Another property of supramolecular polymers is the fact that the supramolecular polymers are self-healing materials. Here you have a film in the middle here. You cut it with scissors, you superimpose the two ends, and you can, after some moment, you can press with your finger and you can stretch and it sticks. So supramolecular polymers lead also to self-healing materials, a very important property also for material science. Let's now go back to the main theme. Of course, I could tell you many other stories. Supramolecular chemistry is now has now been described in sets of volumes of 8-10 volumes, and of course, it's now it's an extremely large field. Like molecular chemistry is a very large field, supramolecular chemistry has also developed into a very large area, and it will continue so. But when we understand better molecular recognition, then we can think of making systems, setting up systems which undergo self-organization. This famous property, which I said at the beginning, is for me the fundamental property of our universe. Our universe has a structure, has a composition, has features which make it able to self-organize. In other words, to generate complexity from simplexity, from simple things, and to lead to very complex organisms like human beings on the planet Earth. It is a sort of a spontaneous way of putting together objects spontaneously, but controlled by their design, by the information which they contain. Let me just show an example, which is a chemical example. It looks like biology, but a virus is not living. A virus is a set of non-living molecules. The tobacco mosaic virus is a virus made of 2,130 protein subunits and one molecule is viral DNA. You see here, these are in some detail, these molecules which form the virus and which can bind to each other side by side with recognition between the surfaces. This is, of course, a self-recognition, this thing. On the left-hand side, you see in a very highly simplified way, the way in which this type of virus builds up. It looks like magic, but it's not magic at all. It is science. It is structural chemistry, organic chemistry, physical chemistry, and it is the way in which this happens, and then on the basis of the way in which these protein molecules making up the virus bind together by their surfaces. It is a programmed chemical system, one may say. Now, this programmed chemical system, the program is molecular, and the operation is supramolecular. The information is stored in the components, and it is processed via the supramolecular interaction patterns. So we can have a look at self-organization. I just want to show you some cases of self-organization as it is done in the laboratory. If we take as components, as bricks, organic molecules which are able to bind the metal ion, and these metal ions will function as connections, cements, between the organic molecules. This has been used extensively, and let me just show you a small selection of objects which have been made in this way. Here you see two artificial helices, a double helix and a triple helix. We made them now many years ago, in the 80s, and they represent a spontaneous formation of a double helix or a triple helix from molecular strands, 2 here and 3 here, and metal ions which hold them together. On the right-hand side, you see the yellow dots here, the yellow spheres, which are here nickel or iron or cobalt ions. Here is a grid made up of 6 molecules, 3 horizontal, 3 vertical, and linked together at each intersection by 9 silver ions. Here is a nanocylinder with 3 linear molecules in red, 4 flat molecules in blue held together by 12 metal ions. This means that when you synthesize the red molecule, the blue ones, and add copper to them, spontaneously this nanocylinder assembles. They have nothing else to do than just letting go. Here are some nice circular architectures which we have already made also over the years. So one can here notice that, in fact, this type of structures, of course, are rather simple, but one can sort of make the assumption that, therefore, programmed self-organization will be of great interest for nanoscience and nanotechnology. Because if one can generate complex architectures spontaneously, but in a controlled way, one can hope that it will be possible also to make systems, to have systems where a device can progressively, successively build up from the components. And this would then be maybe not an alternative, but an excellent complement to nanofabrication, nanomanipulation. In other words, self-organization of artificial entities may lead from the need to make fabrication to self-making, self-fabrication. That's, of course, the ultimate fabrication, which can be of great interest for instance, electronics, informatics, molecular machines, and so on. So, molecular chemistry can be done by design, introducing information to molecules and programming the system so that it can lead to the controlled generation of functional molecular and supramolecular entities. But there's another approach, and the next step in complexity is beyond design, using design, but going beyond, adding another feature, which is selection. And this means that this type of systems will lead to a specific architecture spontaneously, in a controlled way, but based on selection of the right objects which are needed for building up this architecture. For this, you need diversity in the building block to be able to select the good one, the right one, and dynamics so that there's a collision, but the object can dissociate, reassociate, deassociate, and so on and so on. And in the process, it can therefore have features of dynamics in the constitution. I mean here, by this rather complicated word, but it means exactly what it should mean. Constitutional dynamic chemistry is the chemistry of objects which are dynamic in their constitution. They may be dynamic in their molecules and dynamic in their supramolecular entities. Now, if they can dissociate and reassociate, and if the conditions in the environment change, then the molecule can adapt, can hopefully change itself also to adapt to the medium, and leading to adaptive chemistry, which is now what we are mostly doing in my group. So this constitutional dynamics, which I mentioned, can allow for adaptation by variation of the constitution. And this has then been used for searching for biologically active substances, for dynamic nanostructures, for dynamic materials. And we have just seen that also, that self-healing, responsive, adaptive materials can be generated. I has just showed a very, very brief and short example. Let me just give you an example how it functions with biologically active substances. Just that gives you an idea about how constitutional dynamic chemistry operates in the generation of a given selective entity. Suppose you have a set of components shown here with different structures, molecular components with different structures represented here by different colors and so on. And you have here a black dot, which is a functional group capable of performing a reversible reaction. Mixing those together leads to a dynamic combinatorial library constituted of different entities which represent all the possible combinations of the components. Okay, so you can in this way generate all the possible combinations. This is a terrible mixture. So what can you do? What you can do is the following. You add to this a receptor represented here by this binding site here in the receptor. Then the receptor, through molecular recognition, will interact with these constituents shown in the middle. And it will, of course, bind best this or those constituents which fit best the receptor. That means that if we consider that the receptor is the lock, it will bind best the key, the molecule, which fits into the lock. So one can say that the lock, the receptor, assembles its key. The receptor assembles its substrate. This is, of course, in principle, a very powerful mechanism. It's just based on thermodynamics, on generation of a complex, of a more or less complex set of exchanging components which generate constituents, and the addition of the receptor leads to the selective binding of that which fits best. And that is a sort of a dynamic search, can be used as a dynamic search for biologically active substances. That means for drug discovery. We have done a number of those things, but I haven't time to get into it. I wanted just to show you that these dynamic systems have properties which static systems don't have. For static systems, for making a drug, you make many molecules, and you test them all separately or in a mixture, but you test them, the separate molecules. Now, with dynamic chemistry, dynamic covalent chemistry, what is happening is that the system generates all possible combinations, and it's the target which selects among these all possible combinations the one or the ones which fit best, which interact best. Let me just show you an example in material science. I have here to be rather brief, and I cannot give you all the details, but I think you will see what it means, what it amounts to. Suppose we make two polymers with AB and A prime B prime, two components which link together. And the bond between AB and between A prime B prime is a reversible bond. If you superimpose this part, these are dynamic connections, okay? Now, if these are dynamic connections, they can break and reform. So, at the place where they're superimposed, two new combinations can occur, can form AB prime and A prime B. Altogether, there are four possible. AB and A prime B prime both there at the beginning, and then you can generate the mixtures here. If one of these two new constituents has a color, you will see it. Has a fluorescence, you will see it. Now, my Japanese co-workers, then they like these nice pictures. They make here the head of a cat, with the head and the ear is polymer AB. The inside of the ears and the mustache and the eyes are polymer A prime, B prime. You superimpose and you hit, and you see, you get color and fluorescence where it is superimposed. That shows that indeed it works. That means that these bonds can break, then the system can recombine and generate these new features these new constituents which have a color and which have then also a fluorescence. It means that you can really use these dynamic properties to generate properties which do not exist in static materials. So I would like to summarize and then go to some general considerations. Chemistry starts with being molecular. We have to know how to make molecules, of course. That is very, very important. It's the art and science of molecular chemistry. Molecules get together to make populations and have societies of molecules, so to say. This is supramolecular chemistry. And on the basis of the interactions between these molecules, an organization is formed. The molecules have to be dynamic in order to be able to exchange pieces so as to be adaptive, and that is towards more and more complex form of matter, of which living matter is a very complex form. So much for the most chemical part, but let me now reflect a little bit on chemistry and on more general questions. You see here, you see that from what I said, of course, it was very brief, but you nevertheless can remember that objects were made, chemical objects were made, which did not exist before. This is one characteristic of chemistry, to be able to create novel expressions of complex matter. So one can say that the book of chemistry has to be written not only to be read. Of course, we have to read the book of chemistry, the book of what is already generated in the evolution. But nature, evolution, did not produce all possible combinations which chemists can produce. One can also say, in a similar fashion, the score of chemistry must be composed and not only to be played. So chemistry has creative power, which we can represent by, again, on my first slide, when the universe was shown, Big Bang, there was a statue on the right-hand side, which is the thinker of Auguste Rodin. This is another sculpture I show you here of Auguste Rodin, or Rodin, I suppose, in English, where the hand of the sculptor expresses out of a stone a sculpture which was not contained in the stone. This stone does not contain that. It's the hand of the sculptor which expresses it out. And the same way, chemistry is the art of matter, recombining elements of matter to generate new structures, new combinations, which are as creative as what an artist is doing. Now this, speaking of an artist, this is already written down 500 years ago by Leonardo da Vinci. He wrote a really fantastic sentence, where nature finishes to produce its own species. We are part of that. We are one of the species which has been generated by nature in the course of evolution. But then it comes. Man begins using natural things. What are natural things here? The elements of matter, of course. The periodic table of the elements, in harmony with this very nature. What does that mean? That means in harmony with the laws of physics, because you cannot go against the laws which rule our universe. And the end is very strong. Remember that Leonardo da Vinci is a great artist, a great scientist, and a great engineer. This combination gives him a very strong statement to create an infinity of species. In other words, here it is clearly said that man is able to create an infinity of species, most of them are not present in nature, because there are so many to be done, so many to be made. So this is a very strong sentence, which really represents the creativity of chemistry, but also of science altogether, of course. Chemistry is a part of science. Here we come back to the Greek mythology, and I show you here Prometheus, who has stolen the fire of knowledge from the gods, and he runs away with the fire of knowledge in his right hand, looking over his shoulder to see whether the gods are not running after him to catch him before he can give it to mankind, but he gave it to mankind. He gave this to mankind, and here he shows high the fire of knowledge. So that's fine. We get more and more knowledge, but we cannot give it back. We have to realize that what we know, we know. We cannot erase it. We cannot give it back. We have to live with it. We have to learn. We have to become intelligent enough. We have to become responsible enough to use the knowledge which we gain by science. And one may say that the path, our path in our universe, leads us from accumulating knowledge, the quest of knowledge, to controlling our own destiny. We can now be in charge of our destiny, and not just the universe by natural evolution. I would insist on that. Natural evolution now is known. We know more and more about it, and we will, I am convinced, use this to control our own destiny. We have to learn how to do it. It's a very, very deep, very heavy responsibility, but we have no choice. We have that knowledge, we will use it, and we have to learn how to use it. In the past, this using of knowledge and the build-up of knowledge was going from the animal to the human being. In the present, between human beings, the transmission of knowledge is between human beings. What happens in the future? Maybe this, where we shake hands with something we have created. Now, I don't mean that mankind will also only shake hands with robots. That is not what I mean. I just mean that we will modify ourselves. We will, of course, build robots that already exist. At least a number of them are probably still quite primitive, but okay, they exist. But what I want to say here also is that we will modify ourselves. We already have modified ourselves. Some of you had a cataract and now have new lenses in their eyes. Others may have implants replacing their natural teeth by artificial teeth. Others may have some other kind of device in their body. I can even say, pushing it, that if somebody gets an heart transplant, that person cannot say, let's say, to his or her partner, I give you my heart, because it's not your heart anymore. It's the heart from somebody else. So you have to realize that this is happening. We have modified ourselves, and we will continue to do it with better and better means, better and better control. But we have to learn what we want to use it for. Let me finish by citing a mathematician, David Hilbert. David Hilbert, famous mathematician, is buried in Germany, in Göttingen. Here is his tombstone in the cemetery of Göttingen. And there are two sentences here, written here. What are they? Wir müssen wissen. We must know. This is what drives us scientists. That is our mission. that is the mission which is our goal. And the second part is, wir werden wissen. This is the conviction that we will know, and that is what drives us along the way to try to understand, to try to accumulate knowledge, to know, and then, of course, the most important next step is to know for what we want to use this knowledge. It is in our hands now. We have to learn how to use it. Let me finish by showing again here Prometheus holding high the light of science and of knowledge. We will know science shapes the future of humanity. Participate, and that is especially for all the students who are in the midst of their studies. Here we see our planet, a very, very, very small piece of, let's say, dust in our universe. But we are sitting on it. We are on this planet. And what I want to represent by this planet's turning is that science has no borders. Science is all around the world. Science has no politics. Science has no religion. Science has no flags. Science has no borders, no nations. Science is for everybody, and it is true for everybody. And the science we develop on our planet is also valid millions of light years away on other planets where other thinking organisms exist, because the science will be the same. There is no other science. What we learn is valid everywhere. That gives us, of course, scientists a fantastic responsibility. First of all, showing that knowledge and science will drive our world, and it gives us a responsibility of making good use of it. First of all, of acquiring knowledge, and that is truth. This is trying to be truthful. This is the obligation of science. And then we have to learn what use to make of it. So I hope to have presented a general lecture, which has been carrying, of course, over from one topic to another one. But I wanted just to tell you a few different general reflections on science in general and chemistry in particular. Thank you very much for your attention. It's my pleasure to have been able to present to you this talk. Thank you.
Congratulations, JEAN MARIE, on your excellent and always didactic explanation. We have many questions to you. I will read the two first related questions.
Yes, sure, I can answer some. Not 200, of course, but I can, of course, answer some of the questions. I know there are usually many questions because I touch on many different things. But okay, let's start. Let's do some question. Do you want me to remove the screen, or what do you think?
Okay, I will read you. André and Augusto asks you, dark matter have mass and dark energy don't? Professor, do you think that exists chemistry, a different chemistry in dark matter?
Possible. That's possible. I don't know. But in visible matter, we know what is present. There is probably, and it depends also what you call chemistry, there's other physics. and maybe not even, it's not yet sure that it's other physics, because for the moment, one doesn't know whether there is another physics. It is possible. If they are, but for the moment, even dark matter, dark energy, and the type of entities in our universe, they are for the moment explainable with what we already know. But of course, dark matter, dark energy, that can bring very different things. You ask me a question, I am not a cosmologist. I cannot answer that question. What I say is that visible matter is our matter, and we have been able on our planet to understand, and I think there are other planets with people who have understood that, what our universe is made of. This is, I think, quite fantastic. It's unbelievable, in fact. Okay, what else?
Augusto asks also, in some way, possible to have no carbon-based life in the universe?
Possible, you can, yeah, it's not excluded, you cannot exclude it. It is possible. But as you know very well, chemical entities, or let's say the combinations of the elements of the universe, that means of the periodic table, have certain properties, and they are the same everywhere. So if you want to replace carbon-carbon bonds, for instance, by silicon-silicon, that can, in principle, happen. But it needs other conditions, and you want, now maybe it's totally different, but you want probably also to have an evolution, to have something simple which becomes more complex. You have a transmission of the information between parents and children and so on, and offsprings and so on and so on. So this must also be possible. And don't forget that a carbon atom is the same everywhere. An oxygen atom is the same everywhere. So a carbon-carbon bond or a carbon-oxygen bond is the same everywhere. Of course, if it is on a very hot planet, for instance, like Venus, then the carbon-carbon bond does probably not exist because it's too hot, it breaks. But the silicon-silicon bond or silicon-oxygen bond may then be more, maybe more a possibility there. But the bonds, the chemistries, the reactions are the same. It bothers me. I don't like to speak like this because this is sort of restraining the possibilities, the universe. But right now, one cannot think of something else that a carbon-carbon bond is a carbon-carbon bond, point. You cannot get away from that. Huh? And this is also the reason why on our planet, we can know what happens at the end in the vast universe. A hydrogen molecule is something you can see in the universe by its spectroscopic properties. So hydrogen is hydrogen, point. It's not another thing somewhere else. It's hydrogen. Yeah.
A question from Ana Julia. What is the next step that do you expect in the development of supramolecular chemistry?
Yes, I think at the end, I have very quickly spoken about that. This question often comes up. What is the future of supramolecular chemistry? I say, what is the future of molecular chemistry? Supramolecular chemistry is just another part of chemistry. Molecular chemistry is one part, supramolecular chemistry is another one. So, like molecular chemistry has developed over now almost 200 years, and maybe before that, supramolecular chemistry will do that too. So it will develop because it's a natural process, a natural phenomenon. It's present in the universe, in the entities which form the universe. Now, the next step in terms of development, based on, let me just say that, based on molecular chemistry, supramolecular chemistry can develop. based on supramolecular chemistry, other things can develop. For instance, adaptive chemistry is a development of supramolecular chemistry. One can consider it in that way. So I think it will continue, and we have to have clever scientists, clever human beings who can invent the next steps. For the moment, we are still very far from the complexity of a living organism, but we will, once I think we'll understand it, we'll understand how this happens. Let me just give you an example, which I usually use to give a sense of more complexity developing from combinations of objects. A single molecule of water, HOH, cannot freeze, cannot boil. A glass of water can freeze or boil. What's the difference? The glass of water is also water. Okay? So what's the difference? The difference is interactions. So the fact that a glass of water is a supramolecular object, a single molecule of water is a molecular object. And when you mix together lots of molecules, you get a complicated supramolecular space, and this has properties which did not exist on the level of a single molecule. Okay?
Right. William Demos asks you, Professor Lehn, thank you very much for your talk and contribution to supramolecular chemistry. I am a big fan of you. Do you think...
That's nice to hear. How is it going? How is your work going? Going well? You have to let me know a bit more about what you're doing. Scientists, okay, you know, we are not like rock singers or football players. But science is something which really can bring at least the same satisfactions, I would say, much more profound.
Do you think supramolecular chemistry has been extensively investigated, or there is still a lot to be discovered about this subject?
I think of course, it has been very extensively already studied, but molecular chemistry has been there around for a much longer time. Supramolecular chemistry is only about 60 years now. So, okay, in fact, this is also something. Supramolecular chemistry is not something new. It has always existed. There was always water. So there was supramolecular objects. Water is a supramolecular object, as I said. So I think it has already existed, but studying a given area depends on lots of things, on technologies, on the progress of the knowledge. And so, for instance, if you think about what has been done now is in cosmology, for instance, gravitational waves have been discovered. Now, they are there, and they were looked at because Einstein had predicted that this kind of things should exist. So science accumulates, makes hypotheses, which are then confirmed by experiment. Of course, there's a big difference between something which already exists and which you detect, like gravitational waves. Of course, it's a fantastic thing which has happened. It's an incredible achievement. But on the other hand, I sort of would like to insist to convince people that chemistry is really like an art, because you can make things which don't exist yet. This is something which is fundamental to an artist, to make objects by recombining what you have in your hands, the elements, and making all these possible combinations which do not yet exist. And there might be combinations which are absolutely fantastic. Of course, we ourselves as human beings are a fantastic combination already. So maybe it would be good to come back in 1,000 years to see what has happened.
What are the fields within supramolecular chemistry that you think we need to invest the most efforts in the coming years?
Uses, applications.
Yeah.
Yeah, an obvious one, and the most obvious one is, of course, making drugs. Because, as I said, a drug is a key for a molecular biological lock. So a drug, you do something in an organism, you bind to something as selectively as possible. And by this binding, you can influence what is happening. You can maybe amplify the effect or repress it. That's one. New materials also, materials which have properties which present-day materials don't have, but which have then to be made. And there's a lot of work there for engineers, for technologies to develop these materials, which can be of all types, degradable polymers, which we have made some. I have shown you an example of self-healing polymers. And these are other properties, and maybe adaptive polymers, polymers which change shape, which do something when the conditions change. Of course, we have already in metallurgy, we have already memory objects which have an inbuilt memory, like in some metals, which can present memory effects. This is a very interesting thing, just calling it a memory effect in itself. Of course, it's not as complex as memory with the memory we have, but it is already something interesting where an object, once taken out of its where an object can be forced into a given shape, and then if you distort it from that shape, it comes back to the shape. But this is not so surprising when you have a rubber, an elastic. When you have a rubber, you stretch it, and if you then let it go, it comes back to what it was. A rubber, so to say, has memory, which is due to the fact that there are molecules there which you extend, but which have a thermodynamic property which is preferred as distant, and then they come back. It's just doing this. Very simple, but it's a sort of a memory. Now, I think the words have been overused. Memory is something in reality much more complicated than just a rubber which comes back to its same place, or a piece of metal which does the same. Like a spring. A spring, when you have a spring, you push it, you compress it, and you let it go, and it comes back. And you can call that a memory also. Okay. Any other question?
A question from Cynthia. Professor, what message do you leave for the new generation of scientists?
What? Sorry, what?
What message do you leave for the new generation of scientists?
Metals or methods?
Message.
Pardon?
Message.
Metals. No, I don't understand.
What message?
Oh, message. Yeah. Okay, my message, I have already given it. Science shapes the future of humanity. Participate. That is my message. We all depend on science. And I would like, that is also why I like to show at the end the Earth turning. Why the hell do people have wars and kill each other when we are sitting on a ball? And we could do lots of things. I don't understand that. We have such a mission to try to understand what is going on, to try to to improve things and so on. Now people are killing each other. People are, yeah, it's horrible. I mean, I don't understand that. So I think one of the messages is also, of course, as I said, science shapes the future of humanity, but all us scientists should also try, every time we talk with people who are not scientists, to try to have them behave in a rational, in a scientific spirit, looking for truth, looking for reality, looking for openness, looking for preaching the separations between people. So we have to do that, and the spirit of science is really what we need. I don't think that everybody should be a scientist. We need pianists. We need, Nelson Freire just died recently, huh? You know him, the pianist, very famous Brazilian pianist, no? I don't know how you pronounce it. Nelson Freire. You know him? Okay.
Nelson Freire.
Huh? Sorry?
Nelson Freire.
Freire. Okay. All right. Okay. I'm not good at Portuguese. But we need also people like him, like Marta Argerich also, you know, the Argentine lady. We need these people. We need painters. We need people who make wine. We need people who make bread. We need all this. We need people who make buildings. So we need all this. But I think we should all act in the spirit, which is the spirit of science, the basic spirit of science. Voilà.
Other question from Idra. Do you think the next big breakthrough in chemistry is going to be in computational chemistry? To find
in what chemistry?
Computational chemistry.
Computational.
Yes.
Yeah.
Find enzymes.
Pardon? Sorry.
To find specific enzymes, better materials, or even simulate the beginning of life.
Of course, computation chemistry helps, but computation has to be transformed into something in reality. You can compute a possible structure for a drug, but you have then to make the drug. You cannot just eat a computation, eat a result of an equation. You need a drug. If you want materials, yes, computation is very important, and it is done, of course. Predicting the properties of a material, this is going on in companies. Making drugs is done by computation, by simulation. This is done. So it is a very important part, and I think it has still to be improved to make it more powerful, to make it more predictive. But it already exists, and it is part of the whole game. Huh? We did not do too many. We did a lot of computations in the 1970s, theoretical quantum chemical computations. Nowadays, of course, computers are much more powerful. So one will be able to have much better predictions and much better studies helping experimental chemistry to go forward. And so this will happen, it will improve, and is needed. But at the end of the game, you have to make the object. Okay.
A question from Professor Marcus. Is it possible to figure out any type of life without the existence of hydrogen bonding?
Can we figure out what?
Any type of life without the existence of hydrogen bonding.
That's a good point. Yeah, that's a good point. It's a very good point. Knowing the properties of the combination of the elements and the interaction between the molecules, it seems that this type of things must be there. And the hydrogen bond is probably present. Now, I can't say, of course, one could also say, what about water? That's why not always need water, which is hydrogen bond. Maybe yes, but okay. One could imagine a life in liquid ammonia. That is something which is not too different. I was interested in that because I'm considering seeing, can one imagine life in liquid ammonia? Liquid ammonia is probably the best choice. There's also H2S, but H2S has a temperature, has properties which are less good than liquid ammonia. So liquid ammonia could be a possibility. And there are planets with liquid ammonia. Of course, you can also talk about what about liquid ethane or liquid methane. There is liquid methane on the satellites of Jupiter, I think, but it's very, very, very cold. So any chemical process in liquid methane, it will be very difficult to imagine reactions because it's so cold. In liquid methane, everything is frozen almost. So I think ammonia is something. Okay, I guess. Hello. So finally it worked, huh? It took some time, but
Yeah.
it worked.
I guess we can finish here. I don't know if the people in YouTube can hear me, but anyway, in the name of the whole organizing committee, I'm deeply grateful for your, and it's an honor of having you, Professor JEAN MARIE LEHN, here with us today in our jubileum anniversary. So thank you again. It was an honor, a pleasure, and it's I can say it's an opportunity, one in life. I mean, we are all here very proud of the opportunity to hear you, and we can all just say thank you very much for accepting, and also we are sorry for this technical problems in the beginning.
Yeah, but it has been solved and it worked very well afterwards. Yeah. I would also like to thank you for the invitation. I think it's always interesting, and it's important for us, all scientists, to talk with other scientists, and not only with scientists, also to people in the street. I think it's important. That is very important also. And you contribute very much also to knowledge, to discoveries, to teaching, to education. That is important. And you had a famous architect, Oskar Niemeyer, who died at 103, something like that.
Yeah, I guess.
And he was also one of those artists who was able, out of matter, to make things which was his ideas, his imagination. So you have the mission of educating students, trying to have them become good scientists, and convincing them that science is interesting, and even more so, I would say more general, that scientific spirit is very important for all of us.
It's life-changing for all.
Voilà. So thanks very much for the invitation. I wish you another 50 years of great progress. For the next 50 years, I will not be around to give a talk.
But maybe someone from your lab, who knows?
Someone from my lab, yeah, sure, 50 years. I mean, yeah, sure. There are people now who are young people, yeah, if they are now 25, 30 years old, 50 years, that is something they can achieve.
Probably at the time we are not going to talk about or with each other on the screens. Maybe we can just teletransport to Strasbourg and so on, or maybe it would be easier to bring your students to here. So
that would be yes. You know, just to finish on an anecdote which sort of indicates how things happen. I was invited several times to Brazil, but I couldn't come in most several reasons. But I was there once for 5 hours.
Airport.
You know how this happened? In 1992, there was the Rio Congress, the Rio meeting in Rio de Janeiro, ecology, stuff like that. And it happened that the French president, François Mitterrand, invited some people to go with him to Rio. So I went with him with another number of other people. He had the Concorde, the plane. We left Saturday noon, about noon, from Paris. Then he had to be refueled in Dakar, and then it went to Rio. We arrived in Rio. We spent 5 hours there and came back. So we left on Saturday noon. We are back in Paris on Sunday noon.
So it was like a fast tour in Rio.
Yeah, I knew it's very short. I mean, much too short. I would have liked to stay longer, but I was told that if I get out of it, then I am on my own. I have to find a way to get back.
Yeah, a catalyst tour. I mean, very fast.
No, I wanted to mention it just to show that things change. Of course, there are now problems. Because you mentioned teletransportation, teleportation. This is one of those things where then this can happen. Why not? I mean, it has been done with photons and with molecules even, and so we'll see.
There's a sentence that I like pretty much is the impossible is something that was never made before. So who knows?
Yes, exactly. That's a very good sentence. That's also you should tell that to all your students. Impossible is something which has not yet been made. Yeah, I agree. Okay.
So, thank you very much again. I will close our session here. Just wait a little bit more on the Google Meet. We can talk a little bit more, but nothing. Not much. Em português, eu queria agradecer a presença de todo mundo aqui na palestra, tá? Foi uma honra para mim, professor Wanderley, para todo o programa de pós-graduação, a todo mundo que participou aqui no chat. Desculpa novamente o pequeno probleminha que a gente teve no início, tá? Probleminhas aqui tecnológicos. Lembro que agora já vai iniciar a nossa próxima palestra, então vocês vão ter que pular a janela ali no YouTube. E às 7h30 inicia a nossa palestra de encerramento. Então, muito obrigado por todos, pela presença de todos nesse momento memorável aqui para o programa de pós-graduação em Química da UFSC. Então, muito obrigado e até a próxima. Vou finalizando aqui.